Hello, my name is Victor Jayeola. I'm a PhD candidate in the Department of Plant and Microbial Biology at NC State, and I'm in Dr. Kathari's lab. And today I'll be presenting some of the work we've done to elucidate some molecular mechanisms employed by Salmonella to survive on low moisture foods. And particularly for my poster presentation, I'll be looking at a global gene expression analysis of Salmonella contaminating low moisture foods. The ability of Salmonella to survive on low moisture foods is well elucidated, it's well characterized. There are studies that have proven that Salmonella can survive on low moisture foods such as pistachios for up to 3 years, showing less than 3 log reduction in population, depending on the inoculum level. And also epidemiological data suggests that about 80% of outbreaks involving low moisture foods actually linked with low moisture foods involved Salmonella. And low moisture foods in general also accounted for about 22% of all the outbreaks associated with low moisture with Salmonella in the United States. So this indeed alludes to the importance of elucidating the mechanisms employed by Salmonella to adapt on this food and also to persist for a very long time. So the objective of this current study is to understand the transcriptome response of Salmonella contaminating low moisture foods. That is, when Salmonella contaminates low moisture foods and survives for a long period of time, what are the mechanisms, what are the genes employed by this organism that allows it to survive? And we are using RNA sequencing technology to assess all the gene profiles of Salmonella over time, or Salmonella contaminating low moisture foods over time. So for this particular study, we're using dried apples, in-shell pistachios, and cornflakes. And we inoculated this individual fragment of these low moisture foods with our agar-grown inoculum cells that washed twice. We allowed the inoculated fragments to dry until the water activity of the inoculated product approximated that of uninoculated product. And then we store them in the dark at room temperature. And periodically during storage, we would enumerate the population of Salmonella, thus to see how the population is changing based on, is changing really in terms of the time of storage. And also during storage, we extracted total RNA of Salmonella at different time points. And then the RNA were ribodepleted to remove ribosomal genes that may not really inform, may not add any new information. And then we sequenced the RNA samples using Illumina ISIC to generate about 9 to 10 million reads per sample. So specifically, we isolated RNA at 3 hours, so 3 hours after drying, after inoculation, 3 hours after inoculation. So after inoculation, we dried the samples for 3 hours. So we extracted RNA at that time, and then also at 1 week, 5 and 12 weeks. And to the results, as you can see, we started with a population about 9.2 log CFU per fragment. The reason why we used such a high inoculum level is because we wanted to make sure that we have sufficient cells on the fragment at time 2, after 12 weeks, so that we can have sufficient amount of RNA for RNA sequencing. Also, studies have actually shown that in terms of log reduction, there is not much difference between inoculum level of about 9 log to 6 log. So it's typically when we start from inoculation level that is below 6 log that we begin to see marked difference in terms of inoculum size. So this also is still is still possible. The contamination of low moisture foods at that inoculation level is still possible, even in the in the industry. So as we can see, the population of Salmonella decreased gradually on pistachios and cornflakes over time. So we monitored survival up to around 110 days. However, for dried apples, the population decreased below detection point or limit of detection, which was 10 cells per fragment. After about after 63 days, we could not recover. So we did another experiment to get a higher resolution of time to know when actually the population dropped below detection limit, and we found that it was around 43 days. So after 43 days, we could not recover Salmonella on agar plate. However, we continued to extract RNA of Salmonella, even from dried apples, up to like 8 weeks after it failed to grow on plate medium. And what I'm not showing here, but that that I'll talk about at the presentation is we found out that actually, although the cells were not growing on plate, they were still viable. So we did confocal microscopy to visualize the cells directly on apple surfaces. And we found that even after 50 days, about 68% of the cells were still viable. And that accounted for about 10 to 8 cells per fragment. So only about, technically only about 1 log reduction in terms of the population, actually, although they were not growing. And this actually has significant implication in the, in terms of food safety, because then it brings a new perspective into food safety system validations and also assessment of microbiological risk associated with low moisture foods, because conventional or let me say culture-based methods are still the gold standard when it comes to detection of Salmonella. So the Salmonella may be there on the product, but may not be detected. So that brings at least bring about a discussion as to the relevance and the importance of supplementing classical microbiology methods with molecular methods and or microscopic methods, just to make sure that that the cells are dying when they are not growing, and not that they are in a different physiological state that would prevent them from growing on agar media. As in terms of differential expression genes, so RNA gene expression levels at different times, that is at 3 hours, 1 week, 5 and 12 weeks, were compared to the expression levels in the inoculum suspension. So we normalized that, and then we computed differential expressed genes were genes that were at least differentially expressed significantly by 2-fold were considered to be differentially expressed. And as we can see here for dried apples, we have almost instantaneous response in transcriptome. So after 3 hours, about 130 genes were differentially expressed. And then showing that indeed the cells responded to the change in environment very quickly and very rapidly. And what I'm not showing here, but our findings suggest, is that after 1 week though, the transcriptome profile was stable and so that means the profile at the transcriptome at 1 week, 5 weeks, and 12 weeks, they clustered together. So it didn't change over time. But the take-home message here, though, is that the transcriptome response is very quick, and after about a week or so, it kind of stabilizes. And the same observation was made for cornflakes and pistachios. However, for cornflakes and pistachios, we have more genes that were differentially expressed. So here we have about 372 genes that were significantly upregulated, compared to about 93 in apples. So also we're interested to see if the response, to see how it differs among the 3 low moisture foods that we tested, because that's one of the objectives, to see if the mechanism employed by Salmonella to survive on one product is the same as the other. So and these Venn diagrams are showing the distribution of the differentially expressed genes. So the little circle here is for apple, and then pistachios and cornflakes. So I can see after 3 hours, so this 3 hours, after drying for 3 hours. So only about 51 genes were differentially expressed, only about 51 differentially expressed genes were common to all the products. However, there are still some product-specific responses for each of the product that we tested. And also significant to know, important to note is that pistachios, response on pistachios and cornflakes, they share a lot of similarities compared to with dried apples. And we saw the same pattern after 1 week, 5 weeks, and 12 weeks, showing that indeed the response on cornflakes and pistachios are quite similar, although there was still a lot of specificity as to, in terms of the low moisture product. And so we also did an analysis to kind of classify the genes, because we're trying to make sense out of all these genes upregulated, and we found that a lot of stress-induced genes were upregulated, significantly upregulated. And in principle, the hypothesis is genes that are necessary or genes that are required for the adaptation or survival of salmonella on low moisture foods will be upregulated, and genes that are maybe not needed or processes that are not essential to survival probably will be downregulated. So we can see here a lot of genes that were induced. For example, the DNA which is actually involved in protein folding and misfolding. So when when cells are exposed to stress, there's a lot of misfolding of protein in response to that. So they have molecular chaperones that either fold those proteins or they recycle the proteins back to the original pathway so that they can be properly synthesized. We also found a lot of DNA repair proteins, indicating that actually salmonella on these low moisture foods experience substantial DNA damage because the genes that are responsible or that are involved in repair were also highly reduced. We identified a few transporters, and also very importantly, we found some genes that actually were related to virulence. Example of that is the MVPT, which is actually part of the antitoxin system in the plasmid. Actually was first found on a plasmid in Shigella. But we found also that these genes also were significantly upregulated. And perhaps even more surprising actually are the cycles that were downregulated, or the processes that were downregulated. An example of that is the TCA cycle, and cell motility and electron transport chain that involved NADH. And what is important about this, or what is surprising about this, is that TCA cycle and electron transport, they are employed to synthesize ATP. So a TCA cycle basically involves synthesis of ATP from acetyl-CoA, using acetyl-CoA as the substrate. So what this is suggesting to us is that salmonella, so we can speculate, just speculation based on the data that we have, is that under desiccation, salmonella is actually not upregulating ATP synthesis. So it's not trying to make energy. As a matter of fact, it's trying to reduce biosynthesis of ATP. And the acetyl-CoA that it would have used in this cycle to make ATP, it repurposes them into making other molecules, larger molecules, macromolecules, which we are suspecting are required or are necessary to increase the osmolarity of the cell. So basically, because the cell is exposed to a dry environment, instead of the cell contents leaking out, it's actually making other molecules and accumulating them inside the cytoplasm so that it can balance the osmolarity gradient between the cytoplasm and the environment. And also cell motility is significantly downregulated. That is, because they are not necessary. When it's dry, it's not growing, so it doesn't have to move around. So for energy conservation, so what it's doing is that instead of making more energy, it's actually conserving energy. So what I'm also not showing here is that about 17% of the proteins of the genes that were upregulated are involved in ATP binding. So our hypothesis is that it shifts from ATP synthesis to a more effective way of using ATP or using energy, so that it can repurpose the molecule or like acetyl-CoA or short-chain fatty acids, that instead of catabolizing those, repurpose them into manufacturing or biosynthesis rather of a higher molecule substrate. And so our findings reveal some novel genes. These are just a very summary. So there are still other genes and other biological processes that are differentially expressed. So our findings reveal that genes are differentially expressed upon contamination of LMFs and may be mediating survival of salmonella on low moisture foods. We discovered that transcriptome response to desiccation is very quick, very rapid. After 3 hours, we see a lot of genes that were differentially expressed. And also after a while, it's stable. So that actually could give us an insight into why salmonella can survive for such a long time. Because our data suggests that the mechanisms or the biological processes actually needed for long-term survival are synthesized very early when exposed to the stressful environment. And this finding actually is significant, is relevant, not just because it provides new insight into the biology of the biology of Salmonella, or understanding the mechanisms involved in adaptation of Salmonella to low moisture foods. But also, findings like these also could aid development of improved strategies to control Salmonella contaminating low moisture foods. Thank you.